part. So we found that when the cyst died, so this enzyme, so it's no longer present, but then the vesicular fluid inside the cyst then spark, I would say, inflammation, which then lead to development can lead to development of epileptic manifestation. And this has, of course, a therapeutical implication.
This is Immuno Africa, a podcast dedicated to spotlighting African immunology research. Here, we amplify the stories of Africans researching the immune system to tackle the continent's growing burden of infectious and non-communicable diseases. I am Ralph Okete, a biochemist and science communicator in Nigeria keenly interested in research on immune system. On Immuno Africa, I explore and communicate research through the eyes and lips of Africans who do them. Whether you are a seasoned immunologist, a burgeoning researcher, or even an untrained person, I hope the time you spent here rewards you remarkably. Enjoy!
Dr. Fabien Prodjinotho leads us on our first dive into neglected tropical diseases, a group of diseases that affect some of the world's poorest populations. Now a senior scientist in immunology and parasitology at the Technical University of Munich, Germany, Dr. Prodjinotho's journey into neglected tropical diseases research began from his early days as a young scientist in Benin. At the Technical University of Munich, his research focuses on the immunopathology of various neglected tropical diseases and has led him to several notable findings. Worthy of note is his recent elucidation of the mechanisms underlying the epileptic seizures that characterize Neurocysticercosis, a disease caused by tapeworms, which he talks about in this episode.
I'm Fabien Prodjinotho. And I'm a researcher. And right now I'm a senior scientist at the Technical University of Munich, so in Germany. And how did I get here? So I originate from Benin, and then I always have passion into understanding the immunology determining host-parasite interaction, because quite early I believe strongly that learning from this crosstalk can set avenues to develop therapy. And then on this way I did my bachelor and master study in Benin, and then after I have been selected for a scholarship within the German exchange program, so called DAAD. So I got this scholarship, DAAD scholarship, and then I moved to Germany, and I complete my PhD at the University Clinic Bonn, so in Germany, and that was already on also host-parasite interaction, so the immunology of this, and especially the immune regulation by helminth infection and the antibody that they induce. So after I finished my PhD in Bonn, so I moved now to Munich, where am I to continue my career as postdoctoral researcher and senior scientist now in immunology and parasitology at the Technical University of Munich here. So this is basically about me, and then I'm working right now, as I said, a lot in investigation to understand how parasite in general can then immunoregulate the host immune response, and how we can learn from this to develop better treatment for affected individuals in endemic countries. And I also, beside my immunological research, I really I'm quite committed to build capacity in sub-Saharan African context for immunological research. And then we have been organizing 1 week school, but also webinar or lecture or courses on immunological technique, so immunological implication of infectious diseases for young researchers from sub-Saharan Africa. then to help them to understand how powerful immunology is, to understand disease, and then from them contests to move forward diagnostic of those diseases as well. And I do believe that doing so, we can help building capacity in sub-Saharan Africa, but also to build a platform for sub-Saharan Africa network for research in immunology.
Yeah, thank you so much. And I must say you're doing quite a lot apart from being involved in research. It's interesting that you're also giving attention to capacity building and mentorship and teaching. Now you're working on neglected tropical diseases. I never knew like there were certain diseases that had that tag, neglected tropical diseases. So I think I stumbled upon them online and then read so many interesting facts about NTDs, that's neglected tropical diseases. So I know in Africa there's malaria, there is HIV/AIDS, there is tuberculosis with really high incidence, high mortality. I think that's like the three major diseases plaguing the continent at the moment. So why were you drawn to neglected diseases?
So I think you mentioned this well. So those big diseases affecting people in Africa, but beside those, which are quite also, I won't say under investigated. So we have also this group of 20 diseases called neglected tropical diseases, which affect mostly quite poor people in tropical regions. And I think when I was in Benin, and then I grew up in village, so we have people with elephantiasis, for instance, people with big feet. And then usually you see how those people are affected and they are not really accepted in the society. And this is something that I did not at that time believe that this can be due to really infection by parasites. We used to learn that can be due to other things, non-parasitic factors. And then I attended courses on that during my bachelor degree, where I learned that indeed this disease, elephantiasis, lymphatic filariasis, so it's not due to non-parasitic factors, but then really induced and due to parasitic pathogens. So this really sparked my interest into that to further understand how this happened, and what can we learn from that. So this is basically how I start learning or even deeping into neglected tropical disease. And then I found out that indeed it's not only this disease, but then other diseases in the same context affect people, which are quite underestimated, which are not so open. And then beside lymphatic filariasis, you have also so many diseases like schistosomiasis. Beside malaria, or even after malaria, schistosomiasis is the second big tropical infection affecting people in endemic regions. And then I learned, okay, so you still have so many of those neglected tropical diseases affecting people. I said, okay, this is really a field where I want to research, a field where I want to understand what's happening, a field where I want to give more into to understanding this disease. And as you know, disease today, everything originates from immunology of the host. And that is how I get into, so combining already my interest in immunology and then getting known about neglected tropical diseases. So I just, okay, then I go for that because I want to understand this and at least explain to those people, so what is happening here. And this is how my interest into immunology of neglected tropical diseases sparks. And yeah, I'm here now to further investigate into this field.
As stated earlier on, Dr. Fabien Prodjinotho's work on neurocysticercosis, a disease caused by tapeworm, is the focus of this episode, and that is exactly what we delved into next.
So thank you. I mean, so you are talking about tapeworm now, and then here we have important collaboration and funding to investigate, especially infection caused by Taenia solium. And then in this regard, for Taenia solium, so the larvae, the so-called cyst, is the one causing the disease, and this disease is called Neurocysticercosis, and is quite associated. One of the major manifestations of Neurocysticercosis is the development of epileptic seizure. I found out is that when you take the cyst which inhabit in the brain, so you have two parts of the cyst. So you can have a viable cyst which completely immune regulate the host system and then really prevent inflammation. And then I found that indeed an enzyme present in this viable cyst called glutamate dehydrogenase completely then overcomes the immune system by inducing regulatory T cells, which prevent complete inflammation in the brain, but also in the peripheral circulation as well. But then on the other part, so we found that when those cysts died, so this enzyme so is no longer present, but then the vesicular fluid inside the cyst then spark, I would say, inflammation, which then can lead to development of epileptic manifestation. And this has, of course, a therapeutical implication when you know that then there is a molecule inducing or completely preventing inflammation. There is another fluid containing molecule also inducing inflammation. Of course, you will be able to develop a therapeutic against this, or to foster this induction of regulatory T cells to completely block inflammation, or to have TEV inhibitor antibody to block the molecule that induce inflammation in the brain, at least to completely prevent epileptic manifestation in affected people in sub-Saharan Africa, but also in other Andean regions in the world.
My understanding of what the explanation you gave, so the same cysts, depending on the status of the cyst, could promote inflammation or prevent inflammation.
Exactly, depending on the stage. Yeah. So if I'm elaborating a little bit more on that, so basically the cyst itself, so I don't want to get back to the whole life cycle of the parasite, but the most important thing is that this infection is due to really poor hygiene and sanitation condition, because people living with free roaming pigs, and then the life cycle is complete between pigs and humans. So whereby when people eat then undercooked pork meat, so infested with the cyst of the parasite, so they get infected, and then the cyst develop into adult worm, where it produce eggs, and then at the end, so when people with really poor hygiene condition, no toilet, no proper toilet, no sanitation, so they get in contact accidentally with the cyst, with the eggs of the parasite from stool. So this egg then develop into cyst, and then the cyst somehow has a kind of predilection from the brain and then get into the brain. When they get into the brain and then establish in the brain, so they can remain there when they are viable. And this is now where I want to answer your question about the stage of the cyst. So when they are viable, so they can live in the brain of infected people for years, 20 years, even more, without even instigating symptoms. And then those people live in a kind of win-win situation with the cyst, And then this is what we show now that we just published recently, is that indeed this viable cyst, the so-called vesicular cyst, so they have this enzyme which completely then hijack the immune system in the brain. Also, the most important immune cell, the microglia in the brain, and then they reside there, living in win-win situation with the host. But then when it happened that the system died, so then we are now talking about degenerated cysts. When they died, and then this can be due to just natural death because of the aging. They get old and then they died in the brain. And or people, we have this mass drug administration in endemic region for neglected tropical diseases when you use praziquantel to treat people. So the treatment, anti-helmintic treatment, then kill, basically, to say it in simple words, the cyst. And then when the cyst is now killed, the cyst start now to degenerate over different stages. And then this degeneration of the cyst then cause the inflammation which happen in the brain, because the cyst release molecule from the fluid, inside fluid, which then activate different immune cells to produce pro-inflammatory mediators, cytokine, interferon gamma, TNF-alpha, IL-12, IL-1 beta. And then all this inflammation caused by those cytokine in the brain completely disrupt the blood-brain barrier, but also immune cell in the brain, and cause as well dysfunction of the synapse, neurosynapse in the brain. And then this lead to epileptic manifestation. So to be even clear here, so what drives this epileptic manifestation right now is not known, but then certain molecules have been found to be playing an important role in this in the epileptic manifestation. So this is globally what happened. But then, of course, we can get into details, and then I can also give details about how they affect the immune cells and in which sense, in which regard. So this can lead to epileptic manifestation as well.
Thank you for that explanation. Before we go into details, I have this question, because I'm wondering from the explanation you've just provided. So you mentioned a win-win situation. So when these cysts are still viable, they can live in the brain as if they are not harming humans, and humans aren't harming them. So humans are safe. The individual who has this cyst in his or her brain does not experience any epileptic symptoms and all of that. But then when these cysts die, so maybe from drug treatment or through natural means, they release some molecules and all of that could lead to epilepsy. So I'm wondering, is leaving the cyst trying to protect this cyst and keep them viable? Is it safe? Is it actually a safer method? Okay, so that's my question.
Yeah, I got your question. It's a really good question. We have a study ongoing in Tanzania, and then sometime, of course, people ask me this question. So those people are happy with the parasite, right? So they do not develop symptoms. So why do you want to treat people? So, and then to also make it clear, so we only treat people who develop symptom. So, but then coming back to your question, so and this is what is fantastic about host parasite immunology. So when a host and the parasite come together, and then from the immunological angle, and then so what happens is you have three scenarios, right? So you have a scenario, as you said now, in a win-win situation. So which means that the immune system completely compromised with the parasite, and then accept that the parasite then overcomes the immune system and completely immunoregulate the immune system. So this is the first scenario. And this is what happens when viable cysts are present. And then the other scenario is that the immune system then overcome the immune regulation by the parasite, but without inducing chronic inflammation, which means the immune system completely expel the parasite without inflammation. And then we have also this in our study right now, where you can see that people, based on the antibody that they produce against the specific antibody against the parasite, not that they have been affected, but then they expel the parasite without inflammation. But then the last scenario is that both sides then fight back, whereby the immune system of the host then overcome the parasitic immune regulation, but the cause is chronic inflammation. And this is where people develop epilepsy. So now, do you want to treat people who are happy with the parasite? So this is the main question. Although you see that they have parasite in the brain, I usually use to respond to people, you never know when this parasite will die. You never know when the immune regulation by the parasite will be off, and then the immune system fight back, and then chronic inflammation develop. You never know. But the important thing is you cannot treat people who do not develop symptoms, because you can even increase the symptom in those people. But then you have in the certain patient, so they have viable cysts, and then you have also mild symptom like headache. And then most of the people developing this inflammation I'm talking about in epilepsy are people having mix of viable and degenerating cysts in the brain. And this is how the cysts completely block. So I don't want to get into detail, but then the cysts, as you said, so the inflammation is not only due to the presence of the cyst, but also is due to the location of the cyst. So it can be in the parenchyma, the ventricles, but also in extraparenchymal space, where they block completely the cerebrospinal fluid function, and then this cause, of course, intense inflammation once the CSF is no longer playing its role, and then this lead to development of the pathology. So if you have disease and then you are not developing symptom, so we do not treat you, because we can cause another problem to the already problem present. But then when you are having symptom, of course we need to help you, right? So we need to alleviate the burden from the people. So this is why when we diagnose the presence of the parasite into people, given the history and also the symptoms they develop, so we then provide treatment for those people.
Okay. So you only offer treatment to people who are showing symptoms, or maybe those who have. Yeah. So okay, maybe I want to add, before individuals start developing symptoms, are you able to spot or identify those with a higher risk of developing symptoms?
Yeah, I mean it's also a good question. One of the findings that we came across those last year of research into neurocysticercosis is co-infection with HIV, which is quite prevalent in our study. So people having the cysts in the brain, we saw that the more symptoms you develop or this cyst you have, so you have also HIV, and then this can lead to another type of symptom, so iris and so. And then the question is whether the neurocysticercosis infection then help in this way to have HIV, or whether the HIV then predispose people to get neurocysticercosis. But then the important thing is when you have people with symptoms, which mean really headache, strong headache, or epileptic manifestation all the time, or sporadic epileptic manifestation, or regular epileptic manifestation. So you need, of course, to screen them. So we need to do serology to see whether they have antigen or antibody against the cyst or the adult worm, the tinea, as you said. And then after, we need now to screen their brain with a computed tomography scan, which is quite expensive, but then we need to screen their brain to see active infection, and then easily we can correlate this to the symptom that they're having. So we have people on site who are quite devoted to this, to really easily and quickly also pinpoint this classical development of symptom about headache, but also when epilepsy start then to happen, and they know that this endemic region for neurocysticercosis, then they send the people into our study. So we have doctor on site in Tanzania, this is where we are doing the study, who are quite prepared for that when they have this type of symptom, and they know from which region the people come from. So then they occasionally already spot or at least envision which kind of send them to CT scan or also into our study to screen them.
If, like me, you're wondering what these findings mean for neglected tropical diseases research, and particularly for neurocysticercosis, Dr. Prodjinotho has got answers. Let's hear them.
One of the, I would say, major findings that we also identify here, and this is quite important, is that so when you have when the dying cyst, the dead cyst releasing the vesicle, so inside vesicle, then comes in contact with microglia. And this is the fantastic about the story. What happened is that so they induce a quite inflamed potential prototype phenotype in the microglia, and then at the same time they hinder the microglia to phagocytose dead cells. This is one of the major concerns in this disease. So, which means basically the inflammation is caused by this death of the cyst, the vesicular fluid. But then at the same time, inflammation means that you have a lot of dead cells, right? So you inflame in the environment, you have a lot of dead cells. But then the homeostasis, which can come after that, or the restoring of the homeostasis, needs a really clear function and potent function of microglia to remove all those dead cells or inflamed cells. But interestingly, in this context, with the dead cyst, it completely hinders microglia phagocytosis function, which means microglia are inflamed, they produce a lot of inflammatory factors, but then the cells which died in the environment here, and then we are also showing now that the dead cells cause a lot of apoptosis in T cells, CD8 T cells, CD4 T cells as well. So imagine that you have all those dead cells surrounding the dead cyst in the brain. So you have a concept of inflammation in the brain, but then the microglia, the gatekeeper, they are not able to remove those cells. And this is also where from the inflammation, where from the epileptic manifestation stem, because it's completely blocked the synaptic function of those of the neuron there as well. But this has implication, of course. So which means that basically, if you can pinpoint the molecules which are present in this dead cyst, the vesicular fluid, which induce this intense inflammation, then you can block the action of this. You can block the crosstalk or the binding of this molecule onto microglia or the cell, then to induce inflammation. This is the first really important therapeutic implication here. The second one is that so you can have molecule from the dead cyst and molecule from the viable cyst as well coming into the periphery once the blood-brain barrier is disrupted, which means even without going till CT scan, so using CT scan machine, which is extremely expensive, you can pinpoint which stage of the cyst you have in the brain when you have those specific molecules. For instance, the glutamate dehydrogenase. We found only glutamate dehydrogenase in viable cysts. When you have this around in the periphery, so you can say, okay, this means that glutamate dehydrogenase of viable cysts are present. And when we will pinpoint those specific molecules from the dead cells, the vesicular fluid, which then induce inflammation, for instance, also in the periphery, we can, even without going toward CT scan, we can already say, oh, this patient has dead cysts in the brain, and then inflammation can then can come at any time. So this is an important second implication, which means diagnostic in this regard. And then the last one, or I mean, one of the important last one is now the induction of regulatory T cells that we found. And this is completely a novel pathway in developing regulatory T cells. And then we found that this happened via the role of lipid mediators, prostaglandin, but also a cytokine, anti-inflammatory cytokine like IL-10, which are induced by the glutamate dehydrogenase, this enzyme present in viable cysts. So if you can use this enzyme present in viable cysts and then even foster the action of this enzyme somehow in periphery, but also maybe in the brain, so we in that you can implement or at least foster the development of regulatory T cells, which suppress inflammation in the brain, but also in the periphery. And then you can also use those molecules that we for instance recombinantly express. Then in other setting, and then we are planning some in vivo experiment where we can inject this molecule and then foster locally in tissue the development of regulatory T cells in many inflamed tissue. So those are important, and then we are trying now to understand more about this, how we can use those, or whether even other lipid mediator can play a role in this sense, where we have now a big project in regulatory T cells and SFB, as we used to call it in Germany, where we are now we start now investigating this. So those are just three important implications of those data that we recently published. But there are also many other implications depending on from which angle you see the story.
Thank you so much. Yeah, I think that's a lot of applications, both in developing therapies and in the diagnostics. So I think the diagnosing part will really be helpful, because you mentioned that CT scans, of course, are quite expensive. So, and considering the fact that majority of the people who come on with this disease and maybe other NTDs are from poor background and low income background. So if there are cheaper methods to diagnose the condition, then that will give them hope, increase survival, and also mean that it will be easier to tackle the diseases.
You are completely right. Well, I mean, the enzyme that we are talking about, glutamate dehydrogenase. So another finding we came across is that it's not only present in Tenacious cysts. No, it's also present, for instance, in H. polygyrus, which is also kind of helminth parasite. So which means that is not only an enzyme present in Tenacious cysts, but also is present in other helmet parasite as well, which already show beyond neurocysticercosis the implication of this finding about glutamate dehydrogenase from helmet parasite.
Okay, so quickly before we wrap up, I quickly want to just find out from you, because late last month was the World Neglected Tropical Diseases Day, and there's this 2030 roadmap towards eliminating neglected tropical diseases, I think by 90%. What would you say about the progression towards reaching that goal in 2030?
I mean, you nicely framed this. I mean, the WHO is completely committed now to eliminate or to control those diseases in many countries.
Yeah.
And for instance, schistosomiasis is a goal set by the WHO by 2030 to completely eliminate the disease. But I mean, it's a nice goal. It's an important goal. But to reach this goal, so we still have some drawback on the road. So, which imply the treatment mass drug administration in endemic region to control the development of those transmission of the disease. But this also imply, and this is the major concern that I think even we have now in neglected tropical disease, is to diagnose those diseases. Because the earlier or the safer and less expensive, cheaper technology you have to diagnose those diseases, the easier it is for everyone then to get access to those diagnostic materials. And the easier you get in a hit about the epidemiology, current epidemiology of those diseases, at least to treat this region or those region and this region. Because to really get into details, you have mass drug administration, of course, but then how many times per year do you have this in really high endemic region, for instance? Because this is also, financial problem as well. But the WHO is quite committed in this sense to at least in remote region, to give access to those people. And I think another important point is the hygiene conditions, sanitation. So I mean, most of the neglected tropical disease are the risk factor is really poor hygiene and sanitation conditions. People do not have really access to water, to clean water. They do not have toilet, a proper toilet, and they have really poor sanitation conditions. So if we can help those people, I mean, this is a classical thing, right? So if we can help those people having them clean water, to not go to infested river, for instance, and then where they need to get those infested water, getting schistosomiasis, and so, or they have then built like proper toilet for the people to not have contact with potential infected materials. And I think importantly also is to educate those people. So the education of those people, educate them at least so that they know that they have those disease surrounding, or they have those risk factors surrounding. For instance, if you see neurocysticercosis, if you educate well people, they say, oh, please cook well the pork meat that you are using, that you are eating. And then people will further understand this. Okay, if I cook well the pork meat, okay, I won't get infected, for instance. So this is also education, right? Right. So I will just stress more here, as you said, to really reach those goals, the 2030 roadmap. So we need to better understand the immunology of certain disease, infection, certain NTDs, and then just apply directly then to develop new diagnostic tools which are safer, reliable, cheaper to get into endemic region, portable device. So this is what I will stress out about reaching the 2030 roadmap goals.
Yeah, thank you so much for that. The recommendations, I would say. So I love how you involved everybody from the World Health Organization to individuals in local communities. It has to be joint effort. Yeah. And of course, scientists also, like you, are playing their part and putting in all the efforts to make sure we get better diagnostics and therapeutics and have better understanding of the infections, because we need that understanding to make progress. And that brings us to the end of this episode. Why neglected tropical diseases still pose critical health threats to countries across Africa, Asia, and the Americas. We can count on amazing scientists like Dr. Prodjinotho, who are working tirelessly to help us find the easiest, cheapest, and quickest route out of this mess we've been wrapped in. Although 2030 doesn't seem so far away, if we pull our efforts together and support scientists, we may just smash those goals.
Thank you for listening to this episode on Immuno Africa. If you enjoyed it, please feel free to share it with your network. You will learn first about future episodes and get other immunology related updates by following Immuno_Africa or the Immunology in Africa podcast on social media. See you on the next episode. Bye.